We're going to spend time taking a look at the digestive system. So gastroenterology is a medical specialty that deals with the diagnosis and treatment of digestive tract disorders. The digestive tract, also known as the gastrointestinal tract or GI tract, is a very long tube that begins with the mouth, and then to the pharynx, esophagus, stomach, and intestine. A series of interconnected tubes anywhere it's found in the body is known as a tract, and we see the respiratory tract, the urinary tract, and others within the body. In addition to the GI tract, there are accessory organs that work with the GI tract in order to help with digestive activities. And these include the teeth, tongue, salivary glands, liver, gallbladder, and pancreas. There's 4 stages to the processing of food: ingestion, digestion, absorption, and elimination. Ingestion is when you eat food, you take it into the mouth, you chew it up, and then swallow. And then digestion is the enzymatic breakdown of organic compounds. From large organic compounds, your large proteins and carbohydrates and lipids, into smaller compounds that can be absorbed. Absorption is the uptake of nutrients from the intestine into the blood. Elimination is the elimination of undigested material in the food we eat from the body as feces. So just to be certain that everyone understands the term nutrient, a nutrient is any chemical that cells need in order to live out their life. And that would include water, vitamins, mineral ions, amino acids, glucose, etc. Obviously, we have to eat, and if you don't eat, at some point in time, you starve to death. So the chemicals in food are used by cells for growth, to maintain the cells over time, to repair cells, and allow cells to reproduce and make additional cells. About 60, maybe even 70% of the organic content of the food we eat is catabolized during cellular respiration to make ATP. So the organic classes of compounds include the carbohydrates, proteins, lipids, and nucleic acids like DNA and RNA. And 3 of those 4 organic classes are important components of the food we eat. Carbohydrate makes up 50-60% of the organic content of food, and we take in starch from plants, sucrose, that's table sugar. Lactose is in milk and other milk products. Fructose is a very common sweetener, as is corn syrup, which is a mix of glucose and fructose. Protein makes up 10-15% or so of the organic content of our diet. And of course, protein is from both meat and plant sources, and is broken down to the amino acids that cells need. Lipids, or fats and oils, account for 20-30% of our diet. We can take a look at the structures associated with the GI tract and the processing of food, beginning with the mouth. So the mouth is also known as the oral cavity, and it's lined with non-keratinized stratified squamous epithelium. Stratified squamous is the type of lining tissue that you see in areas subjected to friction. Saliva from the salivary glands flows into the mouth by way of ducts to keep the mouth moist pretty much at all times. The palate is the name given to the roof of the mouth, and as we know, it consists of a hard palate and a soft palate. The hard palate is formed by the maxillary and palatine bones. The soft palate behind the hard palate is an arch of skeletal muscle, and the end of the soft palate is known as the uvula. And the uvula of the soft palate swings upward as you swallow to seal off the nasopharynx, and that helps to direct food from the mouth into the pharynx on its way to the esophagus and then to the stomach. The teeth are found within cup-shaped sockets in jaw bones known as alveoli. That's the same name we give to the air sacs, but they're also applied to these cup-shaped sockets that the teeth fit into. The gums cover the jaw bones all the way up to the base of the teeth. The crown of a tooth is the portion of a tooth above the gum line, and it's covered by the hardest substance in the body, that's enamel. This hard substance, enamel, helps to protect the softer parts of the teeth. We add fluoride ions to the city water supply, and fluoride helps to strengthen our enamel, and that helps to prevent tooth decay. A cavity is also known as dental caries, and dental caries is the decay that occurs in response to bacteria within the mouth. Bacteria in the mouth feed off plaque, and plaque is a sticky coating on teeth that comes from the food we eat. Bacteria release enzymes and acids as they feed off the plaque on the surface of teeth, and those enzymes and acids over time erode away the enamel and lead to dental caries. There are several different types of teeth. The teeth in front are called incisors, and they're chisel-shaped. They're specialized for biting and cutting the food we eat. To the side of the incisors would be the canine teeth. They're sharply pointed, and they're specialized to grasp and tear at food. The premolars and molars in the back have flat surfaces that are specialized for crushing and grinding the food we eat. Within the oral cavity is the tongue, and the tongue is made up of 8 skeletal muscles. On the surface of the tongue, towards the back, are taste buds, which contain gustatory cells that can detect 5 basic tastes. So these tastes are our way of sampling food that we eat and deciding whether it's good for us and whether we should swallow. So the 5 basic tastes detected by the gustatory receptors associated with gustatory cells in the taste buds include sweet, salty, umami, sour, and bitter. Sweet tasting chemicals in food generally signal the presence of carbohydrate. Carbohydrate is good. We need it in our diet, and so in general, if something is sweet, we will swallow and eat it. Salty signals the presence of mineral ions like sodium ions and chloride ions and potassium ions, etc. They're essential nutrients, and so in general, we like salty foods and we'll eat them. Umami refers to the beefy taste of meat, and we think that is good as well, and we'll swallow as a result. Sour is associated with the acidic solutions that we like to drink. So most everything that we drink has an acidic pH, like fruit juices, Gatorade, coffee, etc. And in general, sour taste, if it's not too sour, is a signal that the food is okay and it's safe to swallow. Bitter is the only taste that signals the potential presence of poisons that are not good to eat. And often if a food is very bitter, we'll spit it out or reject it. The frenulum is a fold in the lining of the mouth that holds the tongue to the floor of the mouth. Salivary glands are exocrine glands that secrete about a quart, which is equivalent to a liter of saliva, maybe a little more each day, into ducts that come out of the salivary glands and then travel to and open into the mouth. There is a salivation center that's parasympathetic, and it consists of a group of parasympathetic neurons in the medulla oblongata of the brainstem. There are chemoreceptors and pressure receptors embedded within the wall of the mouth. And when stimulated by food in the mouth as you eat, they send impulses into the salivation center, and that stimulates the parasympathetic neurons in the salivation center to send impulses out to stimulate the epithelial cells of the salivary glands to secrete saliva that then flows into the mouth. There's a reflex associated with the salivation center called the cephalic reflex. So it turns out that the sight of food, the taste and the smell of food, along with sounds associated with the time to eat, will trigger thoughts within the cerebrum about food that send impulses to the salivation center, and it in turn will stimulate the production and release of saliva. So this shows the basic structure of a salivary gland. You've got a duct here that opens into the mouth. And if you trace that duct back up into a salivary gland, the duct within the gland will branch into a network of ducts that are lined by epithelial cells that, when stimulated by parasympathetic neurons, will secrete the solution we call saliva. And then saliva flows through this system of interconnected ducts, or this network of ducts, ultimately out of the salivary gland and into the mouth. There are a variety of chemicals in saliva, to include lysozyme, mucus, and a couple of digestive enzymes, salivary amylase and lingual lipase. Lysozyme is an enzyme in saliva. It's also found in the lacrimal fluid that tear glands make that kills bacteria. It kills bacteria in the mouth that can promote tooth decay. So this helps to keep the mouth reasonably sterile. Mucus is a slimy secretion that thickens up saliva. And I'll tell you about each of these two enzymes, beginning with salivary amylase. So salivary amylase is an enzyme that digests starch. So carbohydrate digestion then begins in the mouth. Salivary amylase is a hydrolytic or water-splitting enzyme, and it breaks a polysaccharide called starch into a disaccharide known as maltose. So salivary amylase is active in the mouth with the digestion of starch. Salivary amylase also moves with food down into the stomach, and salivary amylase is active in the stomach for 10 to 15 more minutes at some point in time, because its optimal pH is 7. At some point in time in the stomach, the acidity of the gastric juice denatures salivary amylase, and once denatured, it can no longer catalyze reactions. But the reaction catalyzed by salivary amylase is a hydrolysis reaction, where the enzyme uses water molecules to break the covalent bonds between the glucose molecules that make up starch. Salivary amylase will attack every other bond, and as a result, it breaks this polysaccharide of glucose into maltose. Maltose is a disaccharide consisting of two glucose molecules bonded together. So, if we started with a starch molecule of 1,000 glucose molecules in length, then salivary amylase would break it into 500 maltose molecules, because each maltose molecule consists of two glucose molecules bonded together. Lingual lipase is a hydrolytic enzyme or water splitting enzyme found in saliva, and it's active at acidic pHs, and the optimal pH for lingual lipase is around 4.5. So lingual lipase is produced by the salivary glands, and it's present in saliva. It flows in as saliva flows into the mouth, but lingual lipase really isn't active until the food that it mixes into moves from the mouth into the stomach. So lingual lipase, for the most part, is active in the stomach, where the pH is lower and more favorable to its pH optima of 4.5. And lingual lipase, as an enzyme, breaks down triglycerides, in other words, fats and oils, to free fatty acids and glycerol. The pharynx, also known as your throat, as you know, consists of three sections: the nasopharynx, the oropharynx, and the laryngopharynx. The glottis is an opening on the side of the pharynx into the larynx, or your voice box, and the pharynx leads to the esophagus. So the esophagus is a long tube, probably 10 to 12 inches in length, that goes down through the thoracic cavity, behind the heart, then through the diaphragm, where it opens into the stomach and the abdominopelvic cavity. The esophagus has a 4-layered wall. And the 4 layers would be the mucosa, submucosa, muscularis, and serosa. This same 4-layered wall construction is true not only of the esophagus, but of the stomach and the intestine as well. So the inner layer, or the lining of the esophagus, as well as the stomach and the intestine, is called the mucosa. In the esophagus, the mucosa is, for the most part, stratified squamous epithelium, whereas in the stomach and the intestine, it's simple columnar epithelium. Behind the mucosa is the submucosa that's very vascular, lots of capillary beds, and very rich with areolar and dense irregular connective tissue. The muscularis, as the name implies, has a lot of smooth muscle cells within it, along with elastic connective tissue. And it's these smooth muscle cells that undergo rhythmic contractions known as peristalsis. And these peristaltic contractions help to move food down the esophagus, through the stomach, and down the length of the intestine as well. The outermost layer that covers the surface or outer surface of the esophagus, as well as the stomach and the intestine, is known as the serosa. A sphincter muscle is often, there is a sphincter muscle that acts like a valve made of smooth muscle cells at the base of the esophagus, where it attaches to the stomach, called the gastroesophageal sphincter. And when the gastroesophageal sphincter muscles contract, it clamps down and closes off the esophagus. And that's designed so that food mixing with the gastric juice to produce chyme in the stomach can't regurgitate or reflux out of the stomach back into the lower esophagus. There is a condition known as heartburn, also known as GERD, G-E-R-D, which stands for gastroesophageal reflux disease. Sometimes it's just called acid reflux. And it's caused by a gastroesophageal sphincter muscle that weakens, generally as one gets older. And because the gastroesophageal muscle weakens, it can't clamp down and close off the esophagus where it attaches to the stomach. And as a result, when food is mixing with the gastric juice to form an acidic chyme, occurs, some of the chyme can spurt back up into the lower esophagus, and because of its acidity, it will kill cells that line the lower esophagus, and that creates a burning sensation in the area around the heart, known as heartburn. A portal system is a system of venules and veins that connects to capillary beds. And there's a couple portal systems in the body, and one of them is the hepatic portal system. The hepatic portal system is a system of venules and veins that connect capillaries within the stomach, intestine, pancreas, and spleen to capillaries in the liver. And the consequence with respect to digestive activities is that all of the nutrients that are absorbed out of the small intestine go into capillaries, and then from there, via hepatic portal veins, from the capillaries in the small intestine to capillaries in the liver. So nutrients that get absorbed in the small intestine go first to the liver, where they are processed. The stomach is a bag-like organ under the diaphragm in the abdominal pelvic cavity. The primary function of the stomach is to mix food coming into the stomach with a solution called the gastric juice, and the mixing of food with the gastric juice produces a solution called chyme. There are very powerful peristaltic contractions that occur in the muscularis layer of the stomach that helps in the formation of chyme with this mixing process. And these powerful peristaltic contractions are often called mixing waves. The production of chyme from mixing food with the gastric juice takes 1 to 3 hours, depending on how much you eat. Chyme would be similar to a vomit. So if you've ever vomited or seen vomit from somebody else, and likely you have, then what you're looking at there is essentially chyme that's been thrown up out of the stomach. There is another sphincter muscle called the pyloric sphincter, and it's associated with the pyloric region of the stomach. That's the part of the stomach that attaches to the duodenum of the small intestine. And when chyme is forming in the stomach, the gastroesophageal sphincter is contracted to close off the esophagus, and the pyloric sphincter muscle, which is embedded in the wall of the stomach where it attaches to the small intestine, it stays contracted as well. And that keeps the food and gastric juice mixing unable to escape the stomach for several hours. Once the chyme forms, then the pyloric sphincter muscle will contract and relax, and when it relaxes, it creates an opening that chyme spurts through in volumes of like 10 to 15 mils, maybe a tablespoon from the stomach into the small intestine. So a lot of chyme doesn't ever leave the stomach at once. Instead, the chyme, once it forms, kind of spurts in small volumes from the stomach through the pyloric region of the stomach when the pyloric sphincter muscle relaxes into the duodenum of the small intestine. Along the lining of the stomach are glands called gastric glands, and they're made of epithelial cells. And the gastric glands that line the stomach secrete a watery solution known as the gastric juice. And the gastric juice then flows up and out of the gastric glands into the lumen of the stomach, where it mixes with food to form a solution called chyme. There are 4 types of secretory cells associated with the gastric glands: mucus cells, parietal cells, zymogenic or chief cells, and enteroendocrine cells. The mucus cells secrete a thick, slimy mucus that comes up out of the gastric glands and coats the epithelial cells that line the stomach, and that helps to protect those epithelial cells that line the stomach from the acidity of the gastric juice. The parietal cells secrete hydrochloric acid and a protein called intrinsic factor that I'll abbreviate IF. The zymogenic or chief cells secrete an inactive enzyme called pepsinogen, and the enteroendocrine cells secrete a peptide hormone into the blood called gastrin. I'll talk a little bit more about the parietal zymogenic and the enteroendocrine cells. This shows the basic structure of a gastric gland. So this would be the lumen or open space of the stomach. And this would be an opening along the lining of the stomach that leads into a gastric gland. This opening leads first into what's called a gastric pit, and then the gastric pit leads to a gastric gland. Epithelial cells line the gastric gland, and there's 4 different types. Mucus cells that secrete mucus, parietal cells that secrete hydrochloric acid and intrinsic factor, zymogenic cells, also known as chief cells, that secrete pepsinogen, and the enteroendocrine cells that secrete gastrin into the blood. The mucus, parietal, and zymogenic cells secrete their chemicals into the gastric juice. And then the gastric juice would flow out of the gastric gland, through a gastric pit, and into the lumen of the stomach. Parietal cells are one of the secretory cells of the gastric glands that secrete hydrochloric acid and a protein called intrinsic factor. So hydrochloric acid is secreted by parietal cells, and it makes the gastric juice very, very acidic. So the gastric juice is a very strong acidic solution of hydrochloric acid with a pH of 1 to 2. The parietal cells contain proton pumps in their plasma membranes, as well as transporter proteins for chloride ions. Now, a proton is the same thing as a hydrogen ion. They're one and the same. So this would be a hydrogen atom, and a hydrogen atom has a proton with a positive charge as its nucleus, and the proton with a positive charge is orbited by a single electron. And a proton orbited by an electron is a hydrogen atom. If a hydrogen atom loses its electron, all that's left is the positively charged proton that makes up its nucleus. And a proton, all by itself, is the nucleus of a hydrogen atom, and we refer to that proton without the electron as a hydrogen ion. So a parietal cell has proton or hydrogen ion pumps in its plasma membrane. And the proton pumps move hydrogen ions out of a parietal cell into the lumen of a gastric gland. The transporter proteins for chloride ions also move chloride ions out as well. And the hydrogen ions and the chloride ions in the gastric juice make the gastric juice a very strong solution of hydrochloric acid. The hydrochloric acid in the gastric juice is important for several reasons. One, the strong acidity. I mean, we're talking a pH of close to 1 of the gastric juice kills bacteria, and that helps to sterilize food and make it safe to eat. The hydrochloric acid in the gastric juice helps to soften food up. Hydrochloric acid also reacts with pepsinogen from the zymogenic cells and converts the pepsinogen to pepsin. And this is a reaction that occurs in the lumen of the stomach. So pepsinogen is an inactive enzyme secreted by zymogenic cells, and it's found in the gastric juice. And pepsinogen within the gastric juice will react with hydrochloric acid to convert the pepsinogen to its active form called pepsin. And pepsin is an active enzyme known as a protease. The parietal cells also secrete a protein called intrinsic factor. And intrinsic factor is essential for the absorption of vitamin B12 from the small intestine. So vitamin B12, as you may recall, is essential for the synthesis of the hemoglobin found in red blood cells. In fact, if you have a vitamin B12 deficiency for whatever reason, it will lead to a type of anemia called pernicious anemia. The intrinsic factor from the parietal cells in the gastric juice will bind to vitamin B12 that comes into the stomach with the food you eat. And that forms a complex known as intrinsic factor vitamin B12 complex. And this complex then moves with chyme from the stomach into the small intestine. Vitamin B12 has to be attached to intrinsic factor in this complex in order to move into the epithelial cells that line the small intestine. So the only way to absorb vitamin B12 from the small intestine is if the vitamin B12 is attached to intrinsic factor. The intrinsic factor vitamin B12 complex is absorbed into the epithelial cells that line the small intestine, and from there the vitamin B12 moves out of those cells into the blood. The zymogenic or chief cells secrete an inactive enzyme called pepsinogen into the gastric juice. And within the stomach, pepsinogen reacts with HCl, with hydrochloric acid, and that converts the pepsinogen to an active enzyme called pepsin. Pepsin is a type of enzyme known as a protease. A protease is an enzyme that breaks large proteins into small fragments of that protein. So you take a protein of hundreds of amino acids, and pepsin, which by the way is optimally active at a pH of 1.8, which is the kind of pHs that you see with the gastric juice in the stomach. And pepsin will break this protein down into several small peptides, each about 10 to 50 amino acids in length. So a protease doesn't break a protein down into its individual amino acids. Instead, a protease breaks a large protein down into peptide fragments. Again, each consisting of maybe 10 to 50 amino acids or so. The enteroendocrine cells are also called G cells, where G stands for gastrin. I always call them enteroendocrine cells. The enteroendocrine cells of the gastric glands that line the stomach secrete a peptide hormone called gastrin, and they secrete gastrin into the blood. Gastrin is secreted from the enteroendocrine cells as food comes into the stomach. The function of gastrin is to stimulate the parietal and zymogenic cells to make a gastric juice. And gastrin also stimulates those mixing waves, the powerful peristaltic contractions in the stomach wall that facilitate the mixing of food with the gastric juice to form chyme. There's a group of parasympathetic neurons in the medulla oblongata of the brainstem known as the digestive center. And the axons of these parasympathetic neurons that make up the digestive center go out the vagus nerve, and then out branches off the vagus nerve to the stomach, intestine, liver, gallbladder, and the pancreas. These parasympathetic neurons that come out from the digestive center go out to a variety of digestive organs. They release acetylcholine. These are cholinergic effects. And these parasympathetic neurons from the digestive center, as we'll see, stimulate all digestive processes. There's a couple reflexes associated with the production of the gastric juice that involve the digestive center, the cephalic reflex, which we talked about earlier, and the gastric reflex. the cephalic reflex. This is, again, the sight, smell, the taste, the thought of food, the sound of a dinner bell, all will excite neurons in the cerebrum that then sends stimulatory impulses to the digestive center and the medulla oblongata of the brainstem. The digestive center is a parasympathetic center, and it sends impulses out parasympathetic motor neurons, and their axons travel out the vagus nerve, then along branches, one of which goes to the stomach, and at the level of the stomach, the acetylcholine from the parasympathetic motor neurons will stimulate the gastric gland cells to secrete a gastric juice, stimulate the interior endocrine cells to release gastrin, and that in turn will stimulate mixing waves within the wall of the stomach. The gastric reflex is initiated by food coming into the stomach. And when food comes into the stomach, it will stimulate stretch receptors in the stomach wall. And it will also stimulate chemoreceptors in the stomach wall. And the stimulation of stretch receptors and chemoreceptors as food comes into the stomach results in sensory impulses traveling out the axons from these receptors, in this case into the brainstem where the digestive center is located. And the sensory impulses coming from the stomach wall, from the stretch receptors and the chemoreceptors, will stimulate the parasympathetic neurons in the digestive center, and they in turn send impulses out parasympathetic motor neurons that result in the release of acetylcholine as the neurotransmitter, and the cholinergic effects include an increase in gastric gland production of the gastric juice, an increase in the release of gastrin from the interior endocrine cells, and stimulation of peristaltic mixing waves that help to mix food with the gastric juice in order to form chyme.